All right, so we want to welcome everyone to our fourth, yes, this is our fourth seminar presentation for this semester. Now, a couple of comments. One is last week we had a speaker on sunscreens. This week we have a speaker on melanin. I did not design it this way. It just happened this way. Okay, I just want you guys to know. These guys picked their dates and it just worked out that the melanin seminar came right after the sunscreen seminar. That's one. In terms of some just business stuff here for the seminar, just make sure those of you in Chem 210 and 418 to put your name present. I see that's happening already. So that's good. Also, if you are trying to get co-curricular credit, please, please, please put your name, your ID, co-curricular credit, and we want everyone to be visible at all times. That is students taking the class and those who are trying to get co-curricular credit. We need proof that you are here at all times. All right, so today, our co-host is Justin Corbett, and he is from British Columbia, Canada, and is currently a senior biochemistry major here at Andrews University, Barron Springs, Michigan. In terms of what he wants to do after his degree here at Andrews, he plans to go back to Canada for dental school. And then one of his missions is to provide dental care to underserved communities in British Columbia and Yukon Territory. Outside of school, he has a good time, mountain biking, backcountry skiing, whitewater kayaking, and mountaineering. So he's an outdoors kind of a guy. So Justin, the time is yours now to introduce our speaker for today.
Okay, thank you for that introduction, Dr. Murray. And now I get to introduce Nathan Gianneschi. So Nathan Gianneschi received his Bachelor of Science in Chemistry at the University of Adelaide, Australia in 1999. In 2005, he completed his Ph.D. at Northwestern University following a Dow Chemical postdoc fellowship at the Scripps Research Institute. In 2008, he began his independent career at the University of California, San Diego, where until 2017, he was Teddy Taylor Scholar and Professor of Chemistry and Biochemistry, Nanoengineering, and Material Science and Engineering. In July of 2017, Gianneschi moved his research group to Northwestern University, where he is currently Jacob and Rosaline Cohn Professor of Chemistry, Material Science and Engineering, and Biochemical Engineering. For his work, he has been awarded the NIH Director's New Inventory Award, the NIH Director's Transformative Research Award, and White House Highest Honor of Young Scientists and Engineers, with the President's Early Career Award for Scientists and Engineers. Professor Gianneschi was awarded a Dreyfus Foundation Fellowship, is a Kavli Fellow of the National Academy of Science, a Fellow of the Royal Society of Chemistry, and is an Alfred P. Sloan Foundation Fellow. I'd like to thank you for taking the time to come and present your work to us, and I know we're all looking forward to hearing what you have to say. The time is yours.
Thank you very much. I'm going to share a screen. What I'm going to do first is turn my email off so you don't hear that dinging. Okay, so let's do this. Okay. Thank you very much for the introduction, and thank you for the invitation to speak to you. This is fantastic. And it's awesome seeing everybody come on the camera too. I agree. I'm teaching 2 classes right now in person, and I value the idea that I can see how you're responding is awesome as well. So thanks for that. Let me know if you can see that. Okay, we're all good.
We are good. We are good.
Okay, fantastic. Yeah, I saw that on the schedule. Fantastic coincidence. And I of course, melanin is very important in human skin, but also in many organisms. And in fact, it's a multifunctional material with many other biological functions, and therefore many potential applications and functions for human health, and also for in other areas of technological pursuit. And I'm going to hopefully give you an idea of our take on that, the kind of work that we're interested in doing with it, and a bit about the fun of the material itself, I hope today. So what you see here is an image of a hummingbird. Hummingbirds generally are just awesome animals, obviously, and I think a lot of people have a sort of sense of feeling for them. They're cute, they're fast, and they're these spectacular color displays that they have. This is a particularly excellent photo taken by, as you can see the photo credit down here, of a green hermit hummingbird. What you're looking at here is iridescence that's coming from photonic display of black melanin nanoparticles that are distributed within the keratin fiber network of the feathers of the animal. It's also melanized in the back of its eye, like you and I. There's an absorbent layer on the back of the retina to aid with daytime vision. You can see its beak is melanized, so the black components, the brown components, certainly, but also the iridescent color. This is a really interesting point about melanin. In the optical range that you're viewing it, so I have my big monitor up so I can see it too. So that's why I'm pointing like that. But when you see it, obviously in the photo, you can see it. What you're looking at is the result of, and we'll come back to this a little bit, optical effects that have to do with the black high refractive index, highly absorbent material distributed within a low refractive index, low absorbent keratin or protein-based fiber network. So this is, if you would extract the melanin from its feathers, it would be completely black. It's not a pigment at all. It's what we refer to as a structural coloration. Some of these birds are super iridescent in that they can appear black, completely black head on, and then and then as they turn, they'll become iridescent, or vice versa. We see that in peacock feathers as well, and I'll show you an image of that later. Now, melanin, as I said, is a vital component in many organisms, or at least assists in the performance of biological systems generally. So structural coloration, this is a different photo of a different kind of hummingbird, also highly colored, but again, not with pigment, with purely structural color. Of note, by the way, not all birds do this. So parrots, for example, are pigmented animals, highly colored. Macaws and animals like that are not iridescent. So birds use both kinds of color, as do other insects. The morphology of the particles, which are like these sort of exploded cylindrical Rice Krispie treat type melanin particles, are how the bird achieves this to some extent, and it's a little bit of a complicated story. Humans, of course, we have brown and black and red shades because of melanin and its varieties, its concentration, its density, its morphology in our skin and hair. As I mentioned, it's also on the back of our eyes, and this is an interesting feature of this. You know, the red light, the red eye effect you have in flash photography from humans. You know, if you take a photo of somebody at night with a flash, that red eye, animals that see well at night have a green kind of color to their eye. So foxes and things like that. They have depleted melanin on their retina in order to create internal reflection to give them better night vision. And so the melanin's absorbing to give us day vision. This is why people with albinism, as in a lack of melanin, can sometimes, they suffer from daytime vision because of this problem of reflection within their eyeball. And so melanin's used in other organs, not just in our skin. It's also present in our brains. In fact, melanin itself, including neuromelanin in your brain, is bright by MRI. Actually shown here is a melanoma that's metastasized to the brain. So this is a skin melanoma of a skin cancer of the tissue that produces melanin. and is bright in a T1 contrast image, which is interesting. It's actually part of the reason we got interested in the field. It has interesting thermal properties. So we've shown together with our team members in Ghent, in Belgium, that long distance migratory birds are often have black on the top of their feathers. Next time you see one, like a seagull, you might notice this. They noticed it. I didn't notice this, and they explored this in a wind tunnel where they heated up their wings, the feather of the bird from above whilst in the wind tunnel. And they showed that at least low velocity flight, that heating effect of being hotter on the top of the wing created excess lift. This is relevant potentially to slow flying drones and not so much rapidly flying vehicles that you and I fly around in, but slow flying objects at least may benefit from that. We find it in deep sea fish. So this is nothing to do with UV light, no protection from actinic radiation or the sunlight, in other words. Rather, it's a thermal insulator, we believe. So these predators, this is a little tiny fish, although they look kind of nasty. It's a little tiny thing that can't survive other than in the deep sea, but is completely black in its skin and also its internal organs. We think it's insulating itself from the cold temperatures so that it can move slightly faster than its prey. And just, I love sort of evolutionary biology is an interesting area. It's outside my field, but we work with people in that field. They also think that this color, this black color lets them, when they eat luminescent prey, other bigger predators can't then see the prey within them because it's shielded by them being black. We'll come back to these guys in a second as to why they're black, and I'll give you a little bit of an idea of the structure. One of the more interesting areas of this for people is that they've been found in radiation-resistant fungi, so including in fungi that have been found to grow inside the sarcophagus in Chernobyl, and also on the International Space Station. These organisms are using melanin not to directly absorb the radiation, but to absorb the reactive oxygen species, so the radiolytic products that are produced by radiation. And we'll talk more about why we're interested in that. And really, it gives broadband protection. It also absorbs metals and small molecule toxins, and that'll be a bit of a topic today, both in our skin and also in other organisms, as you can see there depicted in the cartoon. Through synthesis, through polymer synthesis, organic synthesis, and then elucidation of structure-function relationships by really employing a whole range of advanced spectroscopy, electron microscopy methods, and a lot of effort over many years by a lot of people in our labs, we've been able to mimic a lot of these features. So structural pigmented color, which I'll mention briefly. These colored films are entirely black melanin. If you were to shave that off and suspend it in water, it would be a black suspended colloidal solution. So there's no pigments in any of these images. They're all black particles. We've worked out why they have contrast, why they're T1 contrast agents. We've sort of figured out the fundamentals of that. I'll talk a bit about the technology behind using it as a fabric dye that can absorb toxins and why we'd want to do that. And I'll give you a bit of an idea of where we're going in the human health and potentially cosmetic space with these materials. So let's go back in time a little bit. So melanin is, and you can see the dates on the sort of discoveries below. This has sort of spanned the 20th century. An exploration of nature, in other words, in terms of the types of pigments, and of course, many other biological molecules that we discovered in the same time period. These were described in terms of the organisms in which they're found and actually something to do with the biosynthetic pathways. Each of these, as you'll see, are polymeric materials. So these are highly cross-linked solid materials that are derived from amino acids, but through several steps. And so you see tyrosine at the top of the mountain. This is in most organisms, including in fungi and human beings. It's being made in your cells and tissues right now. As I mentioned, not just in the skin, but all over your body. From tyrosine, which is amino acid, to L-DOPA, which you may have heard of. It's used in a variety of different ways, but in particular in Parkinson's disease. Dopamine, which is also a therapeutic molecule used in controlling blood pressure. Cysteine, which is a natural amino acid. These are all combined to generate these different species. Bacteria also make a version of this, which is a little bit unique. And then of interest today as well will be some of these melanins on the right-hand side. These are what so-called Aloe Melanins, as in nitrogen-free. And they have some really interesting, cool properties that we've discovered through polymer chemistry. So this kind of gives you an idea of the history of their discovery. And what we've done over the years and other groups around the world is try to elucidate structure, elucidate the real complexity of these materials, because they're not just simply derived from these species. They're not homopolymers in the sense that they're not just from one monomer, but they're heterogeneous. They're made from many monomers. And in fact, they include proteins and lipids and other things like that in nature. So we're really grappling with some of the more complex kind of biomaterials in this program. To give you an idea of one of the synthetic procedures that we use, and we use many, but to sort of break it down to one of the more common ones that people use in this field, is to take dopamine and then cause it to oxidatively polymerize. So this is we initiate this process by increasing the pH. And we go through these intermediate cyclization reactions ensue, and then cross-linking begins. And the cross-linking reactions are radical reactions as a result of these oxidative conditions. We have two effects then. We have both covalent bond formation between these rings, and you can kind of see some of the dimers and trimeric complexes that are forming. These go on to make tetramers, and remember, they're three-dimensional polymers, so they're highly cross-linked. They're also cross-linked in the supramolecular sense. So beyond the molecule itself, the materials are stacked. So there's pi stacking as well as covalent bond formation. But you end up with this polycatecholate, there are hetero aromatic rings present, catechol groups present, so they can coordinate metals. Just keep those structures in mind as we talk about some of the architectures. What we see by scanning electron microscopy, and I'll show you a lot of images like this in transmission electron microscopy, if we do that kind of synthesis in water at elevated pH, is the spontaneous formation of spheres like this, so spherical nanoparticles. They look really beautiful, obviously, by SEM. And what you're looking at is sort of 100, 200 nanometer objects that spontaneously form, right? So they're spontaneously forming spheres. We can control their size to some extent, their distribution, by controlling the rates of this reaction. So standard kind of seed and growth pathways for nanomaterials. We also, this is a very long story to go into all of this, but we can also control their morphology. We've made what my grad students call walnut particles, which have some interesting properties. We've also been able to make platelet type architectures that mimic, as you'll see in a second, more closely some of the melanosomes that are found in bird feathers, including in hummingbirds. So most of the time, synthetic melanins that I'll show you on the right-hand side have been spheres. That's sort of what we're limited to because of surface energy effects. But through some other methods, some templation approaches, we've been able to break symmetry, which is quite difficult to do. And again, get at some of what really is going on in nature. So Squid Ink, so you may have, if you've ever, you can buy this at the store, you can make Squid Ink pasta, you can play with it yourself. It comes in this variety, it's essentially excreted as a spherical particle. It's actually very similar to the ones we make. But other organisms make all kinds of awesome particles. So hollow rods, all made from the same kind of material, but different shapes, including this guy, which is like a platelet architecture, which I think hopefully you can see is very similar to ones that we're able to now make. So in other words, we can do sort of biomimetic chemistry or natural product synthesis, but now not of small molecules, the way we learn in organic chemistry, maybe some of you are learning, but instead for materials. So sort of natural product synthesis for materials, both to understand the material and to advance technology. So let's look at color for a second. So the birds that I described to you, in particular the iridescent hummingbirds, why are they colored this way? So sun or any light indeed, but actinic radiation, certainly in the photos that I showed you, is incoming, and then it interacts with different layers, both of low refractive index and high refractive index materials. And then depending on the viewing angle, you'll see a different color based on that interference pattern. This can come in the form of straight thin film interference, the way you would see if you have like an oil slick in water on the ground at a gas station, which is highly unstructured, but the same kind of thin film effect, or in photonic crystals that, for example, lead to these spectacular color displays in peacock feathers. If you were to extract, again, the eye of these peacock feathers, they would be completely black particles. So again, none of that is pigment. All of it is because of structural color. So nanoscale and microscale arranged structures in 3 dimensions. In order to mimic that, we've done some fairly primitive things compared to these organisms, right? I would argue they've had a few billion years of a head start, but let's go with that for now. What did we do? So what's going on? Okay, I'll go back a sec, sorry. What's going on here? Essentially, one of the elementary features of this is a combination of low refractive index, high refractive index material. So in other words, materials that don't refract light very much and materials that do, combined with materials that absorb light very heavily. So the melanin is very black and the keratin is not. And that sort of, you can see that in the electron, this is actually electron scattering image, right, by TEM, but you get the idea about electron density. Our attempt at this was to make core shell particles. So what we did was make melanin particles where the core is 160 nanometers in size. And then we grew silica on the surface. So this is our high refractive index core, low refractive index shell. And then we assemble them into films and three-dimensional objects. So what we're doing is holding the core constant and varying the shell so that the distance between the high refractive index material in the assembled objects is variable. And just to give you a brief summary of this, a lot of work over many years in our lab with a lot of collaborators. We've been able to make a whole spectrum of colors. These could be used as printable inks. So we've done inkjet printing with them. We've painted them on surfaces. This is an example of a red-green color blindness test that says NU for Northwestern on it that we published recently. So these are like stabilized inks that could be used to print things. We've done a lot of theoretical exploration with a collaborator, Arthi Jayaraman at Delaware, who's also figured out why they're colored this way and how to predictably generate structural color pigments. So that's a really interesting area of exploration for us. What I'm going to do for the majority of time is pivot to where we find these interesting in mimicking the way melanin works in human skin and how it might be used to protect human skin, both as a preventative measure, so a prophylactic like a sunscreen, or as a material that can block radiation, and then also after injury, the way that it might be used as a healing agent. So melanin in the skin. So in human skin, and indeed in the skin of many other organisms. Another fun fact about melanin is that polar bears, and we've got some photos of shaved polar bears, they're completely black skin. They have clear fur in order to attract light. It appears white to us because of light scattering, not because it has it doesn't have white fur, it has clear fur that directs light down towards its black skin. You notice the nose of a polar bear is black, right? It's helping it absorb sunlight because obviously it lives in cold region. When they hunt seals, they'll actually cover their nose with their hand because they know that, right, the seal's going to see this little black dot coming. So they're both camouflaged. It's kind of ingenious, right? They're both camouflaged and also absorbent at the same time. So human skin can act the same way in that when we upregulate melanin in the skin, and you can see here the micrograph on the left, this is the epidermal layer, and you can see the cartoon for comparison on the right-hand side, filled with keratinocytes, so cells that make keratin fibers. They ultimately die, and then they form this protective layer, a hydrophobic layer of dead cells on the surface of your skin that, you know, when you spill water on your hand, it doesn't go into your bloodstream, right? It rolls off like water off a duck's back. Underlying those layers are melanocytes, relatively low population of melanin-producing cells. And they do something pretty spectacular. They do an unusual thing, actually. They make a particle of melanin, which is made in a organelle called a melanosome. Melanosome is the organelle in which melanin is made. It's unusual in that it's an excreted organelle, right? Nuclei and things like that never get excreted. They're used by the cell. Melanocytes excrete this organelle and keratinocytes pick it up. So keratinocytes in your skin don't make melanin. It's all made by melanocytes. And incidentally, melanocyte cancer is the melanoma cancer I was talking about earlier. So that's what melanoma is. It's a cancer of these cells. And you can have other kinds of skin cancers, of course. What they do when they produce the melanin is excrete it, as I said, and then it gets taken up by keratinocytes. I like this cartoon. It's a little simple, but it puts the point across, right? Excreted organelle gets picked up by keratinocytes, and then it gets deposited around the nucleus as a little cap. And I'll show you another micrograph on the next slide. So this is increasing pigmentation. So fairly light skin, moderately light skin, and dark skin. And you can see in each case, as we melanize, what's happening is and those of other organisms too, you have your nucleus, and then you have a little cap on top of it. That cap is called a perinuclear. It's in the perinuclear space, so a perinuclear cap. So the melanin is in close to the vital nucleic acids. It's also called a microparasol, which I kind of like even better because it's like a little miniature umbrella, right? That your cells have over the top of them, pointing outwards towards the surface. So in other words, sunlight's coming in here. And just to orient, you see this top of the skin that's kind of frayed like that in the micrograph. That's the stratum corneum. That's the protective layer. This is the epidermis. That's full of live cells that are now, in some cases, not every case, but filled with melanin. And then the underlying dermis. this was Dimitri Dayan's a marine biologist at the Scripps Institution for Oceanography, who we work with, who collected the dragonfish, by the way. He also collected these sea limpets, which are awesome. They also become melanized in response to actinic radiation, in other words, sunlight, and they also pack melanin around their nuclei. So it's not just you and I, right? Organisms like this do it. A cousin of this sea limpet that lives under the ocean, where the sunlight can't get to, is completely clear. It doesn't have any need to produce the melanin. It's in no danger of getting skin cancer, right? So it doesn't melanize. So melanin takes some energy to produce, right? We produce it in response to sunlight. We also produce it constitutively in our skin, depending on the color of our skin, in order to protect ourselves from the environment. And also, by the way, our hair is doing the same thing, but so are the sea limpets, which is kind of awesome, right? So what about us? So we've been able to intervene in this process. We had this, well, one of my grad students had this crazy idea. What she said was, and you'll see the video of what she did on the right-hand side, what she had the insight was, Yuran Huang, who's now left my lab and is happily working for a chemical company. In 2017, she said, what if I take melanin particles that I made, so the little spheres that I described to you, and feed them to keratinocytes? Would they get taken up? And I said, no way, this is never going to work. And of course, it does work. And so she was right. On the right-hand side is a video of that process. These are human keratinocytes derived from neonates. So these are from human foreskin, from circumcision in the hospital. So fresh, proper human skin cells, just like yours and mine. What they're doing is growing in the dish, and she's feeding them melanin. They're taking melanin up and becoming tanned. They're becoming melanized. And you can even see the melanin's packing around the surface. You can see, if the video is moving too jittery for you, on the left-hand side, you can see melanin caps that you see in the micrograph on the left-hand side under different staining conditions. We can also see that the cells are very well formed next to each other. So in other words, we have an intracellular suntan. We've shown in other studies then that this protects them. You can kind of squint at this data, but basically what this is saying is, if the cells are melanized by us, so artificially melanized, they're protected from sun damage, from UV damage, which is exactly what you would expect, but highly unusual, right? That an organism would believe that a synthetic particle was natural. In fact, they spread around the nuclei just like the natural melanin. So it's a pretty spectacular example of an artificial organelle. The problem is they don't get into cells unless you culture them in a two-dimensional dish. So it's a pretty good movie, but actually in reality, when we apply it to human skin, and that's what you see up here, they don't get into the skin, right? So what we're doing here, this is from people who've come into the hospital at Northwestern for a tummy tuck. They remove excess skin. And then we're lucky enough to get a hold of it. So people who've donated skin to science, basically it ends up some of the time in our labs. And what we're doing is, in this case, you can see this is a fairly white, non-melanized piece of skin, which is a good control for us. We then apply melanin under different conditions. That's on the left-hand side. So we've applied particles in different creams and agents like that. And then we hit it with UV light. Wherever you see pink in this assay is cell death. So this is cell death caused by radiation. When we treat them with the particles, we see no pink. And so essentially the particles act as a screen. They're not inside the cells, but they're preventing UV light from penetrating the tissue. Now, this is interesting to some extent, but there are lots of other sunscreens. So we got more deeply interested in how this might be applied instead of before you get irradiated, but after you get irradiated, after the skin is damaged. So I'll tell you a little bit about that story. And first, I'm going to tell you a little bit more about melanin itself. So remember that melanin is not just an absorber of light, which it is, it's black, which tells you that that's the case. It also absorbs in the UV, there's no doubt about it. But as I just described, its main predominant activity is radical scavenging. So when the light hits the cell, it produces radicals. Those radicals would damage your nucleic acids and proteins, and melanin is there as a decoy. So it's there as an absorbent of those radicals. And if you've studied free radical chemistry, you know it can be highly productive if done correctly, but usually destructive in a biological environment. So you want to head that off. Organisms like this do this too. So for example, there's a classical example of natural selection that you may have learned about in high school or even in college, where during the late 1800s industrial revolution in England in particular, which led the way, the trees were covered in soot, and the observation was made that a population of speckled moth had shifted to be, instead of white speckled, it was the black ones that had survived, and the darkened melanized one. And the idea was that they were the ones that the starlings, that the birds couldn't see because they were against the tree. Turned out in 2013, not us, but another lab, this is pretty cool. We actually sort of thought of this, but we looked in the literature, they've done it. In fact, the birds have no problem seeing the white ones, the gray ones, the black ones. It's that the black ones can absorb metals, heavy metals and organic toxins that are in the environment from the industrial road. So it's not that evolution didn't happen or natural selection. It's that the mechanism wasn't camouflage. It was absorption of the toxin. So again, not acting as a decoy of reactive oxygen species in this case, but a decoy for heavy metals and for organic molecules. And it's been seen across lots of species. So birds tend to become blacker and more melanized in response to toxins in the environment. Sea snakes do the same thing if they live near human habitation. So they're protecting themselves. So melanization kind of has this superpower, right, of absorbing not just UV, but small molecules, metals, and also bacteria. And I'll tell you about that in a second. This is a really cool experiment where people had noticed anecdotally that people who work with diesel fuel, their skin will darken. This was a controlled study. They took people, they somehow convinced these people to agree to this study where they would apply diesel to their skin. And over time, they saw an upregulation of melanin. The melanin is not produced because of sunlight, right? It's produced to prevent damage from the fuel, from the toxins. Butter clams have been doing this for a long time, more than just in 2021. Butter clams, as they produce this shellfish toxin, they'll upregulate melanin. So the melanin, again, is absorbing its own toxin. So this is an example of an organism that's actually making a toxin to poison other things and has to protect itself by melanizing. Arthropods melanize themselves in order to protect from bacteria. So melanin freshly produced can be made metal-free. And so the idea here is that, well, our thought as well, although this is described differently by others, is that it can be used as an antimicrobial. We've shown it in our lab that fresh melanin, because it's such an excellent chelator of iron, can be used to deplete the environment of iron and therefore reduce the ability of bacterial colonies to form, because bacteria need a source of iron. So it's acting as a material type siderophore. It's also scavenging radicals. So it's also helping the organism deal with the infection itself. So melanin is a broadband absorber, if you like. And so we had this hypothesis, right? We said, okay, if melanin's such a good sponge, does high surface area or porous melanin exist in nature? Or rather, does it act as a really high efficient sponge in nature? That was not known. Still is not exactly known, although we're figuring it out now. And then the next question we had, okay, we'll figure that out later is kind of what we said. In the meantime, can I use synthetic chemistry to make it an even better sponge than it already is? So as I said, if it's a sponge for these things, can I make it more porous and therefore a better sponge? And so one of my grad students, Zofia Sawicka, who's the first author on this paper, also talked me into this. And I originally said it probably wouldn't work, but she did it. She worked really hard and got this to work. So what she did was she took mesoporous silica, and then she templated, so this is silica with nanoscale pores. She grew melanin on the surface, but also inside the silica. And then she etched it with HF, which is, the bone melter. It's a pretty dangerous chemical, which is part of the reason I didn't want to do it. But it works really well. We end up with a porous silica material that's templated, so it has the same porosity or very close porosity to the original porous silica. And I'll come back to these particles in a sec. In parallel, I had a grad student who's also graduated, who was excellent and worked together with two other grad students who graduated as well on Aloe Melanin. So this is the one that I told you about earlier that doesn't contain nitrogen. We worked out how to take this precursor molecule and essentially cross-link it through this radical reaction. So a little bit of organic chemistry for you, of course, right? We generate a radical. That radical can, of course, be spread, obviously, into the ring through resonance, right? So the radical is stabilized by this kind of structure, as you guys know. And, of course, it can then react at these different positions. And so we can make all kinds of dimers, tetramers, trimers, and that's obviously how the cross-linking works. So there's some radical chemistry in action for you. These things, over time, can be made to age differently, and they go from gray to black over time as it cross-links. So it becomes a more and more highly oxidized material, just like when something burns, right? So in this case, what we see, okay, we take this species, we oxidize it, and then we allow it to oxidize over time, 2 days in air, 72 hours in air, and then we can etch these particles by a procedure that doesn't really matter. But we can control its porosity. We can control, this is now an electron micrograph of the materials. We can make them like solid bowling balls. We call them lacy particles, where they're kind of ball spheres with some pockmarks inside them. And then we can make them like ping pong balls, completely hollow inside. And this student wondered, and along with the other ones, how porous are these materials? So on the mimetic, so on our Melanin mimetic, which is the one I described on the template, these are fairly porous materials. So the typical surface areas you expect for something like this, it's not particularly porous, is in the order of tens of meters squared per gram. The higher surface area materials are like hundreds of meters squared per gram, which is non-trivial, that's fairly high. And then the other melanin, the Aloe Melanin version, the nitrogen-free version, is really high. So these are approaching, 900, 1,000 meters squared per gram, which is like the porosity of an engineered material. So we're pretty excited about this at this point. So this is now a couple of years ago, and we published it last year. We probably should have realized this would be the case. If you look at some of the very first papers on polymers of intrinsic microporosity, so these are polymers that already are porous. This was one of the first ones they made. This is a group in the UK, published this in 2004. I wasn't really paying too much attention to that, but I probably should have, because you can see, right, the dimers we make, and obviously the oligomers that probably are being made by the organisms are actually quite similar to these species. Now, different in key ways, but as it turns out, as we've gone on and explored the nature, the natural melanin itself, it actually doesn't look dissimilar to the type of things that humans reinvented in the 21st century. So, right, these organisms have been doing this for billions of years. But one of the questions we have is, why is it, right, that nature evolved to utilize biological polymer of intrinsic microporosity, or what we now call a biopem? So we sort of discovered it, obviously, by the result of, not through chemical intuition, which I wish I could claim, but rather through experimentation, which is not an unreasonable way of doing science. Okay, so what do we do with that? Okay, so we have these porous materials, and we said, okay, well, let's do a demonstration of this. Because melanin's such a good pigment, right? It's such a good pigment for your cells and tissues, as we described. It's in your hair, it's in your skin, it's stuck to keratin, not just in your hair and skin, but obviously the feathers of these birds. Can we take a fabric? So we did it with silk, we did it with wool, so that's a polysaccharide. Silk obviously is a polypeptide. We did it with NICO, which is a nylon cotton weave that's used in military uniforms. This is actually a naval camouflage pattern that you see here. What we did was melanize it. So in other words, we dyed the fabric. And you can see here in my screen, hopefully you can see it. These are darkened, but you can kind of still see the camouflage underneath. When we look at the SEM images, we can actually see that the fibers are intact. They're not frayed, which is nice. So the fabric still has integrity, but it's not, but it's fully intact. So you can see here a zoom out image of the woven fabric, but we've dyed it. As a little segue, of course, right? This is exactly the way that the fibers in our hair and of course those of jaguars and pandas are also colored like that. Of course, the white fur is another story, but that's the way that works. We've also developed this, I'm going to quickly mention, into a hair dye. We're trying to see if we can commercialize this as a replacement for commonly used hair dye components, but we'll see where we go with that. But what we were doing in dyeing fabrics was this. We were asking the question and this is the same kind of fabric dyeing procedure that you see on the top left. And again, the same type of images I showed you earlier. What we were interested in is whether or not melanin would block nerve gas or other toxic molecules, so insecticides, but allow water through. So you can imagine military uniforms, or farmers could wear equipment now that's not oppressively hot, like a plastic coat, or be specially trained to use materials like that, but rather just have clothing that's already naturally absorbent. So that's been our aspiration. So we worked together with some DoD labs to develop this. We published this with them last year. And so what did we get? So this is the data here. So this is a nerve agent simulant that's actually an insecticide, but it's very similar in structure to nerve agents, nerve gas agents. What you're seeing here, you can squint at this, but what basically is going on here is that the layer, you impinge the gas on this, on the fabric itself, and then you look for how much leaks through in time. And basically the melanized fabrics don't let the gas through. The next question we asked is, unlike a straight film, we know how to block these gases, but they have to be like a film that also won't be breathable. So one of the key requirements would be that for a uniform material is that it lets water in and out. And so that's what's shown here. Basically, water can get in and out of the sample, whereas the plastic control down here, which also doesn't let the nerve gas through, is also not breathable. So hence the title of the slide, right? Melanization of the fabrics leads to breathable fabrics that resist toxin breakthrough. And so everything I just said is in the data there. But I'll show you the cartoon. My grad student also made this kind of awesome cartoon of this. So what we can do now is coat fabrics. We're working more heavily now on the textile side to see how far we can push this in blocking toxins, but being breathable. And for the last part of my talk in the next 5 or so minutes, 10 minutes, what I'll tell you about is the next step in this. So these, just like what I showed you in the suntan lotion, let's say, or the UV protection via prophylactic application, this would also be a preventative measure, right? You would put on a piece of clothing that's protective. But what about if you've already been injured, which is the much more realistic scenario, right? Especially for normal civilians and people walking around. And just, by the way, people in the kitchen, right? You're going to get burned in the kitchen. You're going to get a bad sunburn. You'll get a bad thermal burn, or after injury from, say, surgical operations. These are all the same sorts of, or collections of inflammatory instigating insults in the epidermal layers, right? That can lead to other problems, infection, problems of healing, diabetic wounds. So there's a lot of clinical need in this space. And so what we got interested in was whether we could then remediate or deal with post-exposure problems and damage. So I'm going to tell you that story now as the final part of this. Why did we think this would work? Well, I've mentioned reactive oxygen species quenching, or its antioxidant performance. So in other words, it's scavenging activity. This is an assay. This is a colorimetric assay that we do. Not exact. I should have put the mechanism on here for students, so sorry about that. But this is a radical mechanism that gives rise to a colorimetric response as we soak up more of the radicals in solution. Very briefly, what we've got is a porous set of particles and the non-porous, and we show that the porous ones in blue work much better. So less number of micrograms, the antioxidant perform better. That speaks to our hypothesis, right? If I have a wiffle ball that can absorb radicals, it's going to perform better by mass, by microgram, than a completely solid particle, because there's more contact area, right? The radicals can enter the wiffle ball and be quenched. That at least was our hypothesis, and it bears out. So then we went into some animal models for this. We managed to get some money from the NIH to pursue this. So this is now completely in the human health space and potentially in cosmetic applications. But basically, the idea is this. So we take the mouse, we treat it with nitrogen mustard. So nitrogen mustard, you may recall, looks like this. Mustards generally have this structure, right, of a nucleophilic kind of atom in the center of these arms that are alkyl halides. So sulfur mustard looks very similar. It would be sulfur in the center, so a thioether with 2 chloro arms. There are also a whole collection of nitrogen mustard drugs. These were, so unfortunately, this is the story of chemotherapy, right? This is a chemotherapeutic agent. It's an alkylating agent that essentially destroys nucleic acid's ability to propagate. It was used, obviously, in the First World War, as you probably all know, in order to kill people and obviously damage their skin. And we still don't have good remediation methods for these kinds of molecules. So there's both a remediation, there's both an application in chemical weapons as a countermeasure, which we're interested in, but also in the in the healing of wounds that are caused during topical chemotherapy. So there's both a sort of a more like, the chemical weapons angle on that, but also injury from the chemotherapy. And I should say the same sorts of injuries, of course, can occur in response to thermal burns. So straight up burns from heat and UV light. So this is a model system, but also one that might be useful in humans. So what we do is treat the animal with the damaging agent. So that's the treatment, if you like. And then we have the countermeasure. So the countermeasure is the melanin material. And so this at the top is the zoom in of the mouse. So the mouse has been shaved in one spot. The nitrogen mustard's been applied in a gel. And then you see this onset of inflammation. The yellow outline is where the clinicians have said the swelling's occurring. And in the center, you can see the main injury site where the scab starts forming, the scar, as we call it. You can see that after day 14, the fur still hasn't grown back. This animal has black fur and it hasn't recovered. You may sort of realize this as well. If you get a bad injury in your head, of course, it takes time for, and maybe never, right? In scarred tissue where you have hair on your head, it may never grow back. And so that's kind of what you're seeing here, a slow regrowth, which is because of damaged tissue, damaged follicles, but also just overall, the wound site is slower to recover than in the treatment. So treatment and then the countermeasure, which is the therapeutic solid or porous melanin. And so we're now treating, right, at day 1, we're introducing the melanin particles. And after 14 days, I think you can see that we can measure this and we can see that the injury, actually, even at day 2, you can really see in this case, with the swelling has gone way down. We have statistics on the next slide. But what I really like about this is this demonstration that in this case, the fur has now grown back. And so it's obviously recovered faster. Statistics, reveal the fact that, yes, we're reducing skin thickness. So in other words, swelling has been reduced. One of the main indicators of healing that you'll all know from being kids when you've scratched your leg or something playing outside, that when the scab falls off, that's kind of the first sign that things are going well. Well, that's true in the animal model as well. And that's called eschar detachment or scab falling off, as I would say. The clinicians give it this name, of course. The untreated control is at the top. And this is significant, right? You're at about 10 or 11 days when that occurs, whereas at 50%, right? Whereas these are out for a few more days. These are significant wound healing results for a material like this. More importantly than that, real biomarkers are knocked down. And so we see a reduction in superoxide dismutase activity. What is this? This is an enzyme that you naturally have in place to quench radicals. They get overwhelmed and damaged by this nitrogen mustard treatment. And we can recover its activity by essentially helping it to scavenge radicals. And by the way, this is purely from melanin being stuck to the surface. So this is a topical treatment, right? That's having a downstream immune effect, a downstream initial enzymatic recovery, and then ultimately an effect on the immune system that recovers. And this is the last biochemical piece of data that I'll show you, is that really excitingly, these downstream signals for inflammation. This is a matrix metalloproteinase. This is upregulated in all kinds of organ systems. When people have heart attacks, their heart fills with these proteases that try to remodel your tissue. If you were to cut yourself with a scalpel, these would be upregulated in order to control scar formation. So it's a prevalent signal in all of our organs that's indicative of a late downstream upregulation of the inflammatory response. When we treat with melanin, right, as opposed to when we don't, we get this massive reduction, right, in the melanin treated controls. We see this big reduction in downstream inflammation, which kind of proves it biochemically. But I think one of the most exciting results, we've been really lucky to be able to, with the hospital here, to be able to get a hold of these samples from human beings. So these are people who've come in for a tummy tuck or an abdominoplasty. So they've lost weight and they want to remove excess skin. And so they do that, and we get, if they donate it to medicine and research, we get some of the skin. We're kind of signed up to receive the skin, and we make those little patches that I sort of said this earlier, right? Those little skin punches. But now we're looking at a cross section of one of those where we haven't treated it with nitrogen mustard. And you can see here, if you remember earlier, stratum corneum, the epidermis, and the dermal layer. Again, you can see a little bit of melanin here. So that's the natural melanin that was in this person's skin. That is in the person's skin. This is just a punch of their skin. When we treat with the toxin, so this is the nitrogen mustard again, we see this swelling here. In other words, this epidermal dermal separation. So this is like a blistering effect where the top of the skin is coming off in response to the burn. And so that's what it looks like in the micrograph. So again, epidermis, dermis, and then the separation. When we apply the melanin to the surface of this person's skin, so that, remember, this has been removed from the person first. The person's very happily moved on, right? So this is not on their skin, but it is a real human sample. So we've applied we've applied the toxin, then we've applied our treatment afterwards, and we show recovery. Maybe a little bit of separation in this particular piece, and then in this skin down here, we see this beautiful epidermal dermal layer that's completely intact. And again, this is because we're down-regulating this downstream inflammatory response. So we're pretty excited about applying this in clinical applications like this. As I said, after injury, before injury, and seeing where we can go with this, really at the beginning of what we might be able to do. And again, it's not that it's a magic material, right? This is exactly what melanin's doing naturally in your skin. It's defending you from toxic environment, from sunlight, from all of these effects. We're just sort of employing it now because we can understand synthesis, we can understand its structure. We're redeploying melanin, if you like melanin replacement therapy. So we're learning from nature, in other words. So just to backtrack 1 second. So we've learned a lot, as I said, from nature. We look at all the myriad properties that materials have like this. Remember, these aren't proteins, right? They're not as complex as proteins. They're made from several different simple amino acids. So nature is parsimonious, right? It doesn't want to do much. It wants to use as little energy as possible. The fact that it can deploy the same material, maybe in slightly different forms, but to have these multiple functions is really exciting. You think about using a material like this in resource poor areas where you may want radiation resistance, plus you may want it to be a thermal insulator. And this might be on a spaceship going to Mars. It might be in areas where you might want a sunscreen that can defend you from UV light, that doesn't also destroy coral reefs, by the way, which is what some sunscreens do. There's some applications. So obviously we can really learn from that, in a sense, the resource or the energy efficient way that nature goes about producing multifunctional materials. This is the acknowledgements. This has been a big team effort. Marine biologist, Dimitri Dayan, I mentioned him on a slide. Mike Burkhardt, these are all PIs, and I'll show you the names of our students in a second. This is a multi-university research initiative funded by the Air Force to develop these materials. Mike Burkhardt is a synthetic biology expert and chemical biologist at UC San Diego. Arthi is a theory and simulation expert with an expertise in polymer science. Jeff Reinhardt is a bona fide inorganic chemist who worked out a lot of the metal chemistry that I briefly mentioned. Ali Dinjwala is at Akron. He's broadly interested in biomimicry. He's a polymer scientist and engineer, and is also interested in surface chemistry and spectroscopy. Matt Shorkey used to be at Akron and is now in Ghent at the University of Ghent in Belgium. He studies optical effects and structure in color in birds and biomimicry. He's also an avid bird watcher and is friends with the people who took some of the photos of those birds and also has taken some of them himself. We've had lots of people in our group, so 40 plus people, maybe 50 now, have worked on this, from postdocs and grad students, undergrads, all kinds of folks across these different universities. We work with a lot of different government labs, including Air Force, Army, Navy, NASA, in order to develop these materials. Again, thank you to you guys. This is a photograph of my lab. Almost all these people work for me, except these two do not. They're my daughters and my wife. This is at our place a year ago. They definitely did not work for me. But I figured I'd show you what we all look like. And thank you to these agencies. And I'll I'll lead that with thanking Kurt, who's a leader of our NIH-funded program, and a big time collaborator on the human health studies that I shared with you. This is a photo that Matt took of a bird curiously looking at you. That's all melanized material as well. And this is I'll leave you with this one image. This is a non-glazed porcelain plate that was made by a collaborator of ours who's an artist in Belgium. He's going to be in the US actually, working with the Art Institute, hopefully, and Northwestern to develop this. She's interested in natural pigments and how they can be used to create art. And she does a spectacular job. They did all these kinds of all different surfaces and art exhibits using melanin as a pigment or as a paint to create artistic works. Again, that's all structural color. That's not colored at all. That's not pigmented. That's black melanin on a porcelain plate. So I'll leave you with that. Love to take questions. And again, thank you for having me.
Thank you for spending that time with us and explaining your work. I'd like to open up to any of the students or faculty to ask questions.
So I have a question. So do you work on the Do you work with MIT? MIT Institute for Soldier and Nanotechnologies.
We don't. I do know about it, but I don't work with them.
Oh, okay.
Not
They're
currently.
They're competitors then.
No, not at
No
all.
way.
They do different things.
Yeah.
There are a few of those sort of soldier centers. We have a few different Army and Air Force centers at Northwestern that we work with.
Okay.
We sort of have complementary efforts.
Okay.
The more the merrier when solving complicated problems, right? So
That's correct. Yes. Okay, John, you could unmute and ask a question.
Sure. I was just wondering, when you gave the diagram of the fabric kind of helping to not let toxins through, but allow the water to pass through. I was wondering if you've thought of using it for masks with COVID and everything, or just in areas like in California, where air pollution is a big problem and stuff like that, just for filtering, and where and how is water passing through because of the pores are a certain size in the molecule?
yeah, that's exactly right. Yeah. So right, there's a few different reasons. So we also see, for example, ammonia, which is as you know, similar in size. We trap ammonia, but not water. So it's partly pore size and it's partly chemical absorption. So favorable interactions between the melanin and different types of molecules. Melanin is hygroscopic, but it'll fill with water, but then leak it. Whereas the other molecules adhere to it. We've also shown that the other molecules will adhere to it and then we can convert them catalytically to other species. So it's not that it doesn't interact. It's not that it's hydrophobic. It's that it doesn't encapsulate it. The mask idea has come up. We have to do some more studies to see if it's useful. The other thing is, when you talk about textiles and masks and things, it needs to be relatively inexpensive to be scalable or highly specialized. But I think it can be done. We're talking to a number of groups about scale, what scale you would need to be able to get at. This is where, the chemistry hat comes off and the engineering and, production scale hat has to come on, and you start thinking about cost of goods. But it would absolutely work. It's a question of how much better is it than activated charcoal? What advantage do you get over current systems? One of the really cool things about melanin is that it is such a good dye that it also can serve multiple functions. So it's a black fabric that also has these other protective qualities. And so there's also, there's always performance metrics when it comes to translational materials that we have to take account of. But I think you're onto something, and we'd love to look at that.
Other questions? So you did mention, but I don't think you went into it, at least I can't remember that you did, melanin and bacteria. You said it also protects. Yeah.
Right. And I didn't show you the data. I don't have it in the slides, but it's actually quite new. But yeah, it turns out, so this is, I was not aware of this, and apparently other people had been. But it makes sense. So melanin, if you make it in the lab under really nice pristine conditions, we can make it completely iron-free. But as soon as it comes anywhere near iron chloride or even other sources of iron, so serum, we can take, bone kind of broth that has a lot of iron in it. It will soak up iron. So it'll rip iron out of proteins. It's actually it's an excellent scavenger of the metal. And that's because it's this polycatecholate architecture. So it's actually quite efficient at that. So in other words, if you take broth for growing bacteria and feed in the synthetic melanin, it can deplete the iron to enough of a degree that you can't then grow bacteria in it.
Okay.
So it's not that it's an antibiotic against, say, the bacteria already growing. It's that it will compete with the bacteria for the local iron in the environment. So it's possible that's what these organisms are doing, and other arthropods as well. This is the family of, obviously, you can see flies and organisms like that, crabs, etc., spiders. And so the other mechanism that's implied, of course, is the scavenging, that the bacterial infection is also developing the same problem of inflammation. And so they're also countering reactive oxygen species, but we actually think it's probably both.
A crazy idea. What about reaction or interaction with viruses?
Yeah, we don't know that. We don't know that. My guess is it probably absorbs viruses. It tends to be a very adhesive material. So lots of other labs have looked extensively at that, at its ability to I don't know about viruses. It probably has. I probably should know that. But definitely coatings. So it adheres to all kinds of stuff. I kind of quickly mentioned that we can stick it to silica and things like that. So I'd be surprised if it didn't adhere to viruses. It's possible that the organisms use it that way too.
Right. Right.
Maybe. The thing with viruses, you gotta bind them and then kind of stop them from working as well. So that would be a question of whether they can disrupt like envelope viruses and things like that.
Right. Yeah, that'd be very interesting work if someone gets into that.
Yeah, and it would be relatively easy to check with some non-toxic viruses, just to look at their structure to see if they've been destroyed or not. So that would be a fairly straightforward
experiment,
at least initial analysis, right? Not for infectivity, but just for a simple structural analysis. Yeah.
Correct. Hey, Justin, go ahead.
So in the use of melanin in textiles, are you using melanin's structural pigmentation properties, or does that structural pigmentation property get removed when you make it porous to improve its toxin absorption?
That's a good point. So when we do this coating, it's non-structural, right? This is purely blackening the material. So it's purely adsorbent. Yeah, they're not colored the way we've done it. But we can make other kinds of colored coatings. And I briefly mentioned that in those paints that I mentioned that can be different colors. But no, in this case, we haven't done that at all. It's not organized in 3 dimensions. And you can see that in the SEM image here. It's just a bunch of particles stuck to the fabric. So it's purely a black pigment in that case. I mean, the same way it is in these animals or in human hair, right? It'd be cool if we could get like a iridescent hair dye, but we don't know how to do that.
Yeah. So have you collaborated yet with cosmetic companies and stuff, or?
We've talked to some. We're thinking we'll do our own company.
Yeah, exactly.
Yeah, and then that takes 2 different routes, right? Because we really need to get for the clinical work, we need FDA approval. For the use of new pigments, you also need FDA approval. So there's some hurdle to actually getting it into the marketplace. So it's one of those things where you kind of do it yourself, drive the innovation, and then look for partnerships. And so, but we're very open-minded, about that.
Okay. John, you got a question?
Yeah, I was just wondering too. So I know for like a lot of like paints used in art, like they fade over time. And then but in this case, the quality of the paint is related to the structure. So I was wondering like how the structure stays stable across time and with temperature too.
That's really spot on. I mean, intuitively spot on. That's exactly what the fascination has been with structural color, right? Is that, I mean, take these architectures, right? These are printed displays that, to the eye, they don't look particularly colored, but under the microscope, these can look like completely red or yellow or blue arrays, depending on the size of the particles that we use. So that's an example. You would have to physically destroy that. Like you have to actually go in and scratch the surface to change the color. So it won't fade. It doesn't fade as in the pigment getting bleached, which is what happens with colored paint. It has to be destroyed. And so, I mean, just forget about melanin even. If you were to make a paint that were truly structural at scale, it would essentially be fade proof. It wouldn't be abrasion proof. You could destroy it. I mean, you could blow it up, but you wouldn't be able to change its color with light. In fact, melanin gets darker as it gets damaged. Our collaborator Ali Dinjwala at Akron proved that with some melanized films. UV light breaks some bonds and things, but as it breaks, it actually gets blacker. It's highly resilient, actually. So you can imagine there's this thing called Vanta black, which is a kind of structural black. If you've ever seen that, it's pretty awesome.
I've heard about it, yeah.
Yeah, Vanta is there's some other newer versions of this, just incredible materials that you can paint a 3D object with the paint, like a car, and the whole car looks completely flat. Like you can't distinguish the angles on it. So there are some incredible things you can do with structure that create amazing optical effects. So if you have time, look up VANTA black, and you kind of get the idea of an extreme end of what you can do with structure. Again, not particularly beautiful color, right? I mean, it's just a black car. It's not iridescent the way the birds were, but it's just spectacular in a different way.
Yeah.
You wouldn't be able to look up, they painted a BMW like this. It must have cost them a fortune, but it gives you an idea of what you can do. And yeah, that would be resilient to anything but you actually cutting off the paint.
Okay, well, I think we are good. Maybe one last question, but if not, we will go ahead and thank you again. That was, I learned a lot. It was very fascinating, interesting. Yeah, so thank you so very much. You were actually one of my first persons to accept our invitation. And your enthusiasm comes through in your speech, you know, in your talk, and that's really good. So thank you so very much.
Yeah, no, thank you for having me, and thank you for the invite. I also like really interacting with undergrads, and you guys have any questions about research or anything like that, let me know.
Yeah, do you have opportunities during the summer for students?
We do. We actually have multiple REU programs, but we also take undergrads directly into our labs. If these things are of interest, yeah, students can reach out to me.
John?
If I don't respond, it's because I get too many emails, but just persist a little bit. But yeah, no, we absolutely.
John, what you doing next summer?
I actually have next summer free and was helping.
Okay.
So
Okay.
We'll
shoot
see what
me a note. And yeah, so we have REUs through our Nano Center that we run, but also in multiple other areas that relate to this. So so yeah, that goes for everybody. And and of course graduate school, which is the most important.
Yes. Okay, thank you so very much.
Thanks, guys.
Stay in touch. Okay, take care.
You too.
All right, thanks.
Bye.
Bye.